Hey everyone, welcome to For Science Salah Lotfy channel. Today I've got something super exciting for you, a groundbreaking study that's changing the game in the fight against breast cancer. Imagine using bacteria and plants to target tumor cells. Let's dive into this fascinating research. Breast cancer still stands as one of the top causes of cancer-related deaths globally. Conventional treatments like chemotherapy and radiation come with a slew of severe side effects. But this new approach offers an exciting alternative that could revolutionize cancer therapy. So, what exactly are we talking about here? This study examines the use of gamma radiation-attenuated Clostridium sporogenes endospores as immune boosters combined with Boswellic acid, a plant compound known for its anti-inflammatory and anti-cancer properties. The results? Pretty impressive. Key findings include significant suppression of tumor growth, increased apoptosis, or programmed cell death, through Caspase-3 and p53 activation, reduced inflammation markers like TNF-A and VEGF, and an enhanced immune response giving the body a better chance to fight cancer. These results suggest that microbial and plant-based therapies could be the future of oncology, offering a targeted and less toxic treatment option. Now let's talk impact. Beyond the science, this research has major economic and social benefits. First, reducing healthcare costs. Traditional cancer treatments are insanely expensive and often require long-term care. This biological approach could be a more cost-effective alternative. Second, improving quality of life. By minimizing toxicity and side effects, patients could recover faster and with fewer complications. And third, environmental benefits. Unlike chemotherapy drugs, which contribute to medical waste, this approach uses naturally occurring bacteria and plant-derived compounds, making it a more sustainable option. On a global scale, this research holds immense significance. For Egypt and other developing nations, this could make cancer treatment more accessible where healthcare resources are limited. For the international scientific community, these findings lay the groundwork for future clinical trials, potentially influencing cancer treatment worldwide. And for pharmaceutical innovation, it pushes the development of bio-based cancer therapies, reducing our reliance on harsh chemical treatments. This study represents a major leap forward in the fight against cancer, blending microbiology, immunology, and natural medicine to create innovative treatment solutions. What do you think about this research? Could biological cancer therapies become the future of medicine? Share your thoughts in the comments below, and don't forget to like, subscribe, and stay tuned for more scientific breakthroughs here on For Science Salah Lotfy channel. Hey everyone, welcome back to the For Science Salah Lotfy channel. Today I've got something truly groundbreaking for you, a study that dives deep into the world of bacteria and plants to tackle one of the toughest battles in medicine, breast cancer. So, this study was published in the International Scientific Journal of Microbial Biology, and it's titled Impact of Clostridium, Sporogenes endospores and Boswellic acid on breast cancer induced by N-nitroso-N-methylurea, MNU, in female rats. Yeah, it's a mouthful, but trust me, it's worth every word. The research team behind this study is impressive. We're talking about Professor Dr. Hany Ahmed Marghany Mahgoub, a plant biotechnology expert from Al-Azhar University, Dr. Mostafa Ahmed Hussein Elbahnasawy, an immunology specialist also from Al-Azhar University and a postdoctoral fellow at the NIH in the USA, Abdullah Samir Syed Ahmed Mohammed, a budding microbiologist, Dr. Rokhaya El Sayed Maruf Shams El-Din, a biochemistry lecturer from the Egyptian Atomic Energy Authority. These incredible minds came together to push the boundaries of cancer research. Now let's get into the juicy details. They experimented with Clostridium sporogenes, endospores, yes, bacteria, and Boswellic acid, which is derived from plants. Their focus was on how these could impact breast cancer treatment. Here are the key findings. A significant reduction in tumor growth, enhanced immune response and better control of inflammation, increased apoptosis or programmed cell death through the activation of Caspase-3 and p53, improved antioxidant activity, and reduced oxidative stress. This research suggests a new, less toxic approach to cancer therapy that could complement existing treatments and improve patient outcomes. Imagine a treatment that's not only effective but also gentler on the body. Sounds promising, right? So, what do you think about this study? Could this approach revolutionize cancer therapy? Drop your thoughts in the comments below. And hey, if you found this as fascinating as I did, Make sure to like, subscribe, and stay tuned for more amazing scientific discoveries here on For Science Salah Lotfy channel. Thanks for watching. Breast cancer, a formidable foe, casts a long shadow over countless lives. It is a diagnosis that brings with it a whirlwind of emotions, from fear and anxiety to hope and resilience. It is a relentless disease, often striking without warning and leaving a trail of fear and uncertainty in its wake. The impact is not just physical but also deeply emotional, affecting patients and their loved ones. Despite significant advancements in medical science, traditional treatments like chemotherapy and radiation therapy can inflict harsh side effects, underscoring the urgent need for innovative and less toxic approaches. These treatments, while life-saving, often come with a heavy toll on the patient's quality of life. The search for a cure continues, fueled by the hope of a brighter future for those whose lives have been touched by this devastating illness. The statistics are sobering, yet they also drive the relentless pursuit of better solutions. Breast cancer is the most common cancer among women globally, accounting for a significant proportion of new cancer cases and cancer-related deaths each year. This reality underscores the importance of awareness, early detection, and continuous research. While early detection and treatment have improved survival rates, the battle against breast cancer is far from over. Survivors often face ongoing challenges, including the risk of recurrence and the long-term effects of treatment. The quest for novel therapeutic strategies, particularly those that are effective yet gentle on the body, remains a top priority for researchers and clinicians worldwide. Their dedication and innovation offer a beacon of hope for a future where breast cancer can be more effectively managed and ultimately eradicated. In the realm of cancer research, a groundbreaking approach is emerging, one that harnesses the power of bacterial endospores and natural compounds to combat tumor growth. This innovative method is not only promising but also represents a significant shift in how we think about cancer treatment. This innovative strategy centers around Clostridium sporogenes, a harmless bacterium found in soil known for its unique properties and potential in medical applications, and Boswellic acid, a natural extract derived from the Boswellia serrata tree, commonly known as Indian frankincense. This resin has been used for centuries in traditional medicine for its healing properties. This unlikely pairing has shown remarkable promise in preclinical studies, paving the way for a potential paradigm shift in cancer treatment. The results have been nothing short of extraordinary. The rationale behind this approach is both elegant and intriguing, combining the strengths of both biological and chemical agents. Clostridium sporogenes possesses a unique ability to selectively germinate and proliferate within the oxygen-deprived environment of solid tumors, making it an ideal candidate for targeted therapy. This anaerobic bacterium, harmless to healthy tissues, acts as a Trojan horse, delivering therapeutic agents directly to the tumor microenvironment. This method ensures that the treatment is both effective and minimally invasive. Boswellic acid, with its potent anti-inflammatory and anti-cancer properties, complements this strategy by further inhibiting tumor growth and bolstering the body's immune response. Together, they form a powerful duo in the fight against cancer. To investigate the synergistic effects of Clostridium sporogenes and Boswellic acid on breast cancer, researchers conducted a rigorous preclinical study using a rat model. The study involved dividing rats with induced breast tumors into different treatment groups. One group received a combination of Clostridium sporogenes spores and Boswellic acid, while control groups received either Clostridium sporogenes spores alone, Boswellic acid alone, or a placebo. Throughout the study, tumor size, immune responses, and markers of inflammation and oxidative stress were closely monitored. The researchers employed a range of sophisticated techniques including tumor volume measurements, flow cytometry, ELISA assays, and biochemical analyses to meticulously assess the efficacy and safety of the treatment. This comprehensive approach allowed for a thorough evaluation of the combined therapy's impact on various aspects of tumor progression and the host's physiological response. Clostridium sporogenes, an anaerobic bacterium commonly found in soil, harbors a unique characteristic that makes it a compelling candidate for cancer therapy. This bacterium thrives in oxygen-deprived environments, such as the interior of solid tumors, while remaining harmless to healthy, oxygen-rich tissues. This selective targeting ability stems from the fact that Clostridium sporogenes can only germinate and proliferate in the absence of oxygen. When introduced into the bloodstream, the bacterial spores remain dormant until they encounter the hypoxic or oxygen-deficient regions within tumors. Once inside the tumor microenvironment, the spores germinate, multiplying rapidly and releasing therapeutic agents that can directly attack cancer cells. This targeted approach minimizes damage to surrounding healthy tissues, potentially reducing the harsh side effects often associated with conventional cancer treatments. Section 5. Boswellic acid, nature's defense. Derived from the gum resin of the Boswellia serrata tree, Boswellic acid is a natural compound with a long history of medicinal use, particularly in traditional Ayurvedic medicine. Its potent anti-inflammatory and analgesic properties have been recognized for centuries, and modern science is now uncovering its remarkable potential in the fight against cancer. Boswellic acid exerts its anti-cancer effects through multiple mechanisms. It interferes with the signaling pathways that promote tumor cell growth, survival, and metastasis. Moreover, it inhibits angiogenesis, the formation of new blood vessels that tumors rely on for nourishment and oxygen supply, effectively starving the tumor. Additionally, boswellic acid has been shown to modulate the immune system, enhancing the activity of natural killer cells and cytotoxic T lymphocytes, key players in the body's defense against cancer. Section 6. Promising outcomes, shrinking tumors and boosting immunity. The preclinical study yielded highly promising results, demonstrating the remarkable synergistic effects of Clostridium, sporogenes and boswellic acid in combating breast cancer. The combination therapy led to a significant reduction in tumor growth compared to the control groups. Furthermore, the treatment significantly enhanced the immune response against the tumor. The researchers observed a marked increase in the infiltration of immune cells such as natural killer cells and cytotoxic T lymphocytes into the tumor microenvironment. This heightened immune response plays a crucial role in destroying cancer cells and preventing tumor recurrence. The study provided compelling evidence that the combination therapy not only directly targets tumor cells but also empowers the body's own immune system to mount a more effective anti-tumor response. Section 7. Taming the storm, inflammation and oxidative stress. Chronic inflammation and oxidative stress are increasingly recognized as key drivers of cancer development and progression. These processes not only initiate but also sustain the growth and spread of tumors. Inflammation can create a favorable environment for tumor growth, providing the necessary signals and nutrients that cancer cells need to thrive. Meanwhile, oxidative stress damages DNA and other cellular components, potentially contributing to mutations that lead to cancer. This damage can disrupt normal cellular functions and promote malignant transformation. The combination therapy, in addition to its direct anti-tumor effects, also demonstrated a remarkable ability to dampen inflammation and oxidative stress in the tumor microenvironment. This dual action is crucial for its effectiveness. The researchers observed a significant reduction in the levels of pro-inflammatory cytokines, which are signaling molecules that promote inflammation. These cytokines, when present in high levels, can exacerbate the inflammatory response within the tumor. This decrease in inflammation helps to create a less hospitable environment for tumor growth and survival, making it harder for cancer cells to proliferate. Moreover, the treatment effectively reduced oxidative stress by scavenging free radicals, which are highly reactive molecules that can cause extensive cellular damage. The therapy also boosted the levels of antioxidant enzymes, which play a critical role in neutralizing free radicals and protecting cells. This reduction in oxidative stress helps to protect healthy cells from damage, thereby maintaining their normal function and integrity. Ultimately, this comprehensive approach may contribute to the overall efficacy of the therapy, offering a promising avenue for future cancer treatments. Section 8. A glimpse into the future personalized cancer care. The remarkable findings of this preclinical study offer a beacon of hope for the future of cancer treatment. The combination of Clostridium sporogenes and boswellic acid represents a novel therapeutic strategy with the potential to revolutionize the way we approach cancer care. This innovative approach holds particular promise for personalized medicine, tailoring treatments to individual patients based on their unique tumor characteristics and genetic makeup. Imagine a future where cancer treatment is not a one-size-fits-all approach, but rather a highly individualized regimen designed to target the specific vulnerabilities of each patient's tumor. The ability of Clostridium sporogenes to selectively target tumor cells, combined with the potent anti-cancer properties of Boswellic acid, opens up exciting possibilities for developing personalized and targeted cancer therapies. Section 9. A beacon of hope, the power of innovation. The preclinical study investigating the synergistic effects of Clostridium sporogenes and Boswellic acid on breast cancer represents a significant leap forward in the quest for more effective, less toxic cancer treatments. This groundbreaking approach, harnessing the power of nature and cutting-edge science, has the potential to transform the landscape of cancer therapy. As we delve deeper into the intricate mechanisms of cancer and explore the vast potential of the natural world, we move closer to a future where cancer is no longer a dreaded diagnosis, but a manageable condition. This study serves as a powerful reminder of the importance of continued investment in scientific research and innovation. It is through such endeavors that we unlock the secrets to defeating this complex disease and offer renewed hope to millions worldwide. We stand on the precipice of a new age in cancer treatment, where the boundaries of science and nature blur. Imagine a future where our own bodies, aided by nature's ancient defenses, can fight back against this formidable foe. This vision is becoming a reality as researchers delve deeper into the symbiotic relationship between bacteria and plant-based compounds. This is the promise offered by a recent study exploring the combined power of Clostridium sporogenes, endospores, and Boswellic acid against breast cancer. These natural elements, when combined, show a remarkable ability to target and destroy cancer cells. This research, like a beacon in the darkness, offers a glimmer of hope. It suggests that we might be on the verge of a breakthrough that could change the way we approach cancer treatment forever. It delves into the microscopic world, where bacterial spores and plant extracts hold unexpected power. These tiny agents, often overlooked, could be the key to unlocking new, less invasive treatments. The study's findings, though preliminary, hint at a future where cancer treatment becomes less about harsh chemicals and more about harnessing the body's innate healing abilities. This approach could lead to treatments that are not only more effective but also gentler on the patient. This is not just about treating a disease, it's about understanding the complex interplay of life itself. By studying these interactions, we gain insights into the fundamental processes that govern health and disease. It's about recognizing that even in the smallest, seemingly insignificant corners of our world, powerful forces are at work, forces that we can learn to harness for the betterment of humanity. This research is a testament to the potential that lies within the natural world, waiting to be discovered and utilized. The heart of this research lies in the precise combination of two seemingly disparate elements, bacterial endospores and plant-derived Boswellic acid. This unique blend aims to harness the natural properties of both components to create a powerful therapeutic agent. Clostridium sporogenes, a common bacterium found in soil, possesses a unique ability to germinate specifically within oxygen-deprived environments, like those found in the core of solid tumors. This characteristic makes it an ideal candidate for targeting cancer cells that thrive in such hypoxic conditions. Boswellic acid, extracted from the resin of the Boswellia serrata tree, has been used for centuries in traditional medicine for its anti-inflammatory properties. Modern science is now uncovering its potential in cancer therapy, adding a new dimension to its medicinal use. In this study, it serves as a potent ally, working in concert with the bacterial spores to target and dismantle cancerous cells. The synergy between these two elements could pave the way for innovative treatments. The researchers employed an arsenal of scientific tools and techniques, ensuring accuracy and reproducibility. From advanced imaging technologies to sophisticated analytical methods, every step was meticulously planned and executed. High-precision scales measured out minute quantities of chemicals, ensuring that each component was used in the exact proportions required for the experiments, while specialized incubators maintained optimal conditions for bacterial growth and experimentation. These controlled environments were crucial for the success of the study. This meticulous attention to detail, a hallmark of good science, forms the bedrock upon which reliable results are built. Every observation, every measurement, and every experiment was carefully documented to ensure the integrity of the research. Before being deployed in the fight against cancer, the Clostridium sporogenes endospores undergo a rigorous preparation process. These hardy spores, capable of withstanding harsh conditions, are first carefully cultivated in a nutrient-rich broth, allowing them to multiply and thrive. To ensure safety, the spores are then subjected to a measured dose of irradiation. This critical step eliminates any risk of unwanted bacterial growth, while preserving the spore's ability to germinate within the tumor environment. This delicate balance between potency and safety is crucial. It highlights the researchers' commitment to harnessing the power of these bacterial allies while minimizing potential risks. This meticulous preparation process underscores the study's commitment to both scientific rigor and ethical considerations. To study the effectiveness of this novel treatment, researchers induced breast cancer in laboratory rats. This process involves carefully introducing cancerous cells into the rats, allowing scientists to observe the progression of the disease in a controlled environment. This decision, while ethically complex, is a necessary step in understanding and combating this devastating disease. The ethical considerations are immense, but the potential benefits for human health are too significant to ignore. The rats, bred specifically for research purposes, were treated with the utmost care and respect throughout the study. Researchers ensured that the animals' living conditions were optimal, providing them with a comfortable and stress-free environment. The induction process itself was carefully controlled, with tumor growth closely monitored to minimize any potential suffering. Advanced imaging techniques and regular health checks were employed to track the progression of the disease. This approach, while difficult, reflects the delicate balance between advancing scientific knowledge and ensuring the humane treatment of animals. Researchers are constantly aware of the ethical implications and strive to maintain this balance. It's important to remember that these animals are not just subjects in an experiment, they are stand-ins for countless human lives affected by cancer. Their role in research is crucial and deeply respected by the scientific community. Their sacrifice, while regrettable, paves the way for future breakthroughs that could one day alleviate the suffering of millions. The knowledge gained from these studies is invaluable and holds the promise of new life-saving treatments.
We stand on the precipice of a new era in medicine. Scientists are exploring the microscopic world to combat cancer. This is a story of innovation, harnessing nature to fight disease. Imagine a microscopic warrior, a spore targeting cancer cells. This is the promise of Clostridium sporogenes endospores. Combined with Boswellic acid, they offer a novel approach. This therapy has shown remarkable results in studies. It offers hope for millions battling this disease, a testament to scientific inquiry and nature's potential. The fight against cancer is a marathon, not a sprint. Every step forward brings us closer to a cure. This research is a significant stride in the right direction. It offers a glimpse into a future where cancer is manageable. The success of this innovative treatment hinges on the viability of the Clostridium sporogenes endospores. These microscopic warriors must remain dormant until they reach the oxygen-deprived environment within tumors. To ensure this, scientists employ a technique called serial dilution. This method allows researchers to isolate and cultivate a pure and potent strain of endospores. Imagine taking a single drop of a highly concentrated solution and diluting it in a series of steps. With each dilution, the concentration decreases, eventually leading to a solution containing only a few individual spores. These isolated spores are then grown in a nutrient-rich environment, ensuring that only the healthiest and most viable spores multiply. This meticulous process is crucial for the effectiveness of the treatment. By ensuring the viability of the endospores, scientists can maximize the number of active agents reaching the tumor site. This in turn enhances the therapeutic potential of this groundbreaking cancer therapy. Cancer cells, in their uncontrolled growth, wreak havoc on the body's delicate balance. They generate a barrage of harmful molecules called reactive oxygen species, creating a state of oxidative stress. This oxidative stress damages healthy cells and fuels tumor growth. The combination of Clostridium sporogenes, endospores and Boswellic acid works in part by mitigating this oxidative storm. Researchers measure this effect by analyzing levels of malondialdehyde, MDA, a marker of oxidative damage, and glutathione, GSH, a powerful antioxidant. In studies, this treatment significantly reduced MDA levels, indicating a decrease in oxidative stress. Moreover, the treatment boosted GSH levels, bolstering the body's natural antioxidant defenses. This two-pronged approach, reducing harmful molecules and increasing beneficial ones, helps to restore balance within the body. It tips the scales in favor of healthy cells and hinders tumor progression. The immune system is our body's natural defense force, constantly patrolling for and eliminating threats. Cancer cells, however, are masters of disguise, often evading detection by the immune system. This new treatment strategy not only targets cancer cells directly, but also empowers the immune system to join the fight. One way it achieves this is by modulating the levels of nitric oxide, a NO, and hydrogen peroxide, H2O2. These molecules play crucial roles in the immune response. NO, produced by immune cells, acts as a signaling molecule, alerting other immune cells to the presence of invaders. H2O2, on the other hand, is a potent weapon that immune cells use to directly destroy pathogens and cancerous cells. Studies have shown that this innovative therapy increases the production of both NO and H2O2 in the tumor microenvironment. This enhanced immune response helps to create a hostile environment for cancer cells, making them more susceptible to destruction. Section 5. Vital organs, vital signs. Seeing the improvement. Cancer and its treatment can take a toll on the body, particularly vital organs like the kidneys and liver. These organs play essential roles in filtering toxins and maintaining overall health. Remarkably, the combination of Clostridium sporogenes, endospores and Boswellic acid not only targets cancer cells but also demonstrates protective effects on these vital organs. Researchers monitor kidney and liver function by measuring specific biomarkers in the blood. Elevated levels of these biomarkers often indicate organ damage or stress. In preclinical studies, this novel therapy significantly reduced the levels of these biomarkers, suggesting improved kidney and liver function. This finding is particularly encouraging as it suggests that this treatment option may offer a more comprehensive approach to cancer care. It not only attacks the cancer itself, but also helps to mitigate the adverse effects of the disease and its treatment on the body, improving overall health and well-being. Section 6. Calming the storm inflammation and immune response. Inflammation, while a natural part of the body's healing process, can be a double-edged sword. Chronic inflammation creates an environment conducive to cancer development and progression. Therefore, controlling inflammation is crucial in cancer therapy. This novel treatment strategy exhibits remarkable anti-inflammatory effects, further contributing to its therapeutic potential. Researchers assess inflammation by measuring the levels of specific molecules involved in the inflammatory response. These include NF-κB, a protein complex that regulates immune and inflammatory responses, TNF-A, a pro-inflammatory molecule, VEGF, a protein that promotes blood vessel formation, crucial for tumor growth, and IFNG, a molecule involved in immune regulation. Studies have shown that this treatment significantly reduces the levels of NF-κB, TNF-A, and VEGF. This indicates a reduction in inflammation and a suppression of blood vessel formation, effectively cutting off the tumor's supply lines. Additionally, the treatment modulates IFNG levels, further demonstrating its ability to fine-tune the immune response. Section 7. The dance of death, apoptosis and cancer cell demise. Every cell in our body has a predetermined lifespan. When a cell becomes damaged or is no longer needed, it undergoes a process of programmed cell death called apoptosis. Cancer cells, however, are adept at evading apoptosis, allowing them to divide and proliferate uncontrollably. This innovative treatment strategy exploits this vulnerability by reactivating the apoptotic pathway within cancer cells. It achieves this by increasing the activity of caspase-3, an enzyme crucial for the execution phase of apoptosis, and p53, a protein known as the guardian of the genome due to its role in preventing tumor formation. By increasing caspase-3 and p53 activity, this treatment effectively pushes cancer cells towards self-destruction. This targeted approach ensures that cancerous cells are eliminated while minimizing damage to surrounding healthy tissue, a hallmark of effective cancer therapy. Section 8. A picture speaks 1,000 words. Histopathological evidence. While molecular analysis provides valuable insights into the mechanisms of action, visual confirmation is crucial to understanding the tangible effects of this treatment on tumor tissue. Histopathological examination, the microscopic study of diseased tissue, offers this visual evidence. Tumor samples from treated subjects reveal a stark contrast compared to untreated controls. The treated tumors show significant shrinkage with large areas of dead or dying cancer cells. This is a testament to the effectiveness of the treatment in directly killing cancer cells. Furthermore, the examination reveals a marked decrease in blood vessels within the treated tumors. This reduction in vascularization starves the tumor of nutrients and oxygen, further inhibiting its growth and progression. These visual confirmations underscore the potency of this novel treatment strategy. Section 9. Supercharging the immune system, JAK/STAT3 and IL-2/IL-10 pathways. The immune system is a complex network of cells and signaling pathways that work in concert to protect the body. The JAK/STAT3 and IL-2/IL-10 pathways are two critical components of this intricate system, playing vital roles in regulating immune responses. The JK/STAT3 pathway, when overactive, can contribute to tumor development and immune suppression. This groundbreaking treatment effectively inhibits this pathway, dampening its tumor-promoting effects and boosting the immune system's ability to fight back. Conversely, the IL-2/IL-10 pathway is essential for maintaining immune balance. This treatment enhances this pathway, promoting the activity of immune cells involved in tumor suppression. By modulating these two crucial pathways, this treatment effectively tips the balance in favor of the immune system, empowering it to mount a more effective attack against cancer cells. Section 10, a beacon of hope, the future of cancer treatment. The fight against cancer is a global endeavor, a testament to human ingenuity and resilience. Groundbreaking research on Clostridium, sporogenes and Boswellic acid offers hope for a future where cancer is manageable. While still in preclinical stages, the results are promising. This therapy targets cancer cells, reduces oxidative stress, and enhances immune responses. It also protects vital organs. If successful in human trials, it could revolutionize cancer therapy. The future of cancer treatment lies in innovative, nature-inspired approaches. This research showcases the power of collaboration and commitment to improving human health. Continued research could transform millions of lives, ushering in a new era of hope. The universe of cancer research is vast and ever-expanding. Like celestial bodies locked in a cosmic dance, scientists and researchers are constantly seeking new treatments. The goal is to eradicate this devastating disease. Every day, new discoveries are made. These discoveries ignite hope for a future free from cancer's grip. One such beacon of hope shines brightly from a surprising source, the world of bacteria. Clostridium sporogenes, a common bacterium found in soil, holds a secret weapon. This weapon is its endospores. These endospores, under specific conditions, can target and attack cancer cells, but that's not all. In a remarkable synergy, scientists have coupled these endospores with a natural compound found in the Boswellia tree, Boswellic acid. This potent combination is showing incredible promise in the fight against breast cancer. This is a story of cutting-edge science. It's a story of hope. It's a story about the potential to revolutionize cancer treatment as we know it. Imagine your cells as bustling cities. Energy is generated, resources are transported, and information is exchanged. But just like any city, cells produce waste. This waste, known as reactive oxygen species or ROS, can damage the city's infrastructure if not managed properly. Oxidative stress occurs when there's an imbalance. Too much ROS overwhelms the cell's natural defenses, like a garbage strike that throws the city into chaos. This cellular stress is linked to various diseases, including cancer. One of the main culprits in this cellular chaos is lipid peroxidation. This process damages cell membranes, like rust eating away at a metal fence. One way to measure this damage is by looking at malondialdehyde or MDA levels, a marker of lipid peroxidation. High MDA levels signal increased oxidative stress. This is where our dynamic duo comes in. Clostridium sporogenes, endospores and Boswellic acid work together to combat oxidative stress. They act like efficient waste management and repair crews, reducing MDA levels and restoring order to the cellular city. They also boost the production of glutathione or GSH, a powerful antioxidant. Think of GSH as the city's fire department, quenching the flames of oxidative stress. Reactive oxygen species, or ROS, and their counterparts, reactive nitrogen species, or RNS, are like powerful forces of nature within our cells. In controlled amounts, they play crucial roles in cellular communication and function. However, when cancer emerges, it's like a storm bruise, unleashing a torrent of ROS and RNS. This deluge damages healthy cells and fuels tumor growth. Nitric oxide, or NO, and hydrogen peroxide, or H2O2, two key players in this storm, are often found at elevated levels in cancer. Studies have shown that treatment with Clostridium, sporogenes, endospores and Boswellic acid can significantly reduce NO and H2O2 levels. It's like the eye of the storm. The treatments bring a sense of calm, mitigating the damaging effects of these reactive molecules. Our kidneys and liver are the body's filtration system, working tirelessly to remove toxins and waste products from the bloodstream. Cancer and its treatments can take a toll on these vital organs, often leading to complications. Think of it this way: if cancer is a traffic jam in the body, then chemotherapy and radiation are like using a jackhammer to clear the road. While effective, these treatments can also damage the surrounding infrastructure. Data shows that treatment with Clostridium, sporogenes, endospores, and Boswellic acid can significantly reduce the toxicity associated with cancer and its conventional treatments. It's like using a more precise tool to clear the road, minimizing damage to the surrounding structures. This protective effect helps to maintain healthy kidney and liver function, reducing the burden on these vital organs. Section 5. Inflammation, a double-edged sword. Inflammation is the body's natural defense mechanism, like a team of first responders rushing to the site of an injury. It's a crucial part of the healing process. However, chronic inflammation can be detrimental, like a fire alarm that won't stop ringing. It can damage healthy tissues and contribute to cancer development. In the fight against cancer, it's essential to find the right balance of inflammation. We need enough to activate the immune system, but not so much that it harms healthy cells. This is where Clostridium, sporogenes, endospores, and Boswellic acid demonstrate their remarkable ability to modulate the inflammatory response. These treatments act like skilled mediators, calming the inflammatory storm without completely extinguishing the fire. They do this by influencing key inflammatory markers such as NF-κB, TNF-A, VEGF, and IFNG. Visualize it like turning down the volume knobs on each of these inflammatory signals. Section 6. Apoptosis, commanding cancer cells to self-destruct. Apoptosis is a natural process of programmed cell death. It's like a self-destruct button that eliminates damaged or unwanted cells, preventing them from wreaking havoc in the body. Cancer cells are masters of disguise. They evade apoptosis, multiplying uncontrollably and forming tumors. One of the goals of cancer therapy is to reactivate this self-destruct button, forcing cancer cells to commit suicide. And here's the exciting part. Clostridium, sporogenes, endospores, and Boswellic acid have shown the ability to do just that. They increase the levels of caspase-3 and p53, key proteins involved in apoptosis. It's like reprogramming the cancer cells, reminding them of their self-destruct function. Section 7. The JAK-STAT pathway, a cellular communication highway. Imagine the JAK-STAT pathway as a cellular communication highway. It's a complex network of signals that control cell growth, survival, and differentiation. In many cancers, this highway is hijacked, leading to uncontrolled cell proliferation and tumor formation. Clostridium, sporogenes, endospores, and Boswellic acid act like traffic controllers, restoring order to this crucial communication pathway. They modulate the JAK-STAT pathway, preventing the transmission of signals that promote cancer growth. Section 8. Cytokine power, boosting the immune system's army. Cytokines are the messengers of the immune system, like tiny commanders directing the body's defenses. They play a critical role in recognizing and destroying cancer cells. IL-2 and IL-10 are two such cytokines with opposing roles. IL-2 is like a battle cry, rallying the immune system's troops to attack cancer cells. IL-10, on the other hand, acts as a peacekeeper, preventing an overactive immune response that could harm healthy tissues. Studies have shown that treatment with Clostridium sporogenes, endospores and Boswellic acid can shift the balance of these cytokines. They boost IL-2 levels, enhancing the immune system's ability to fight cancer, while modulating IL-10 levels to prevent an excessive inflammatory response. Section 9, a glimpse through the microscope, histopathological evidence. The proof, as they say, is in the pudding, or in this case, in the tissue samples. Histopathology, the study of diseased tissues under a microscope, provides visual confirmation of the treatment's effectiveness. Imagine peering through the lens of a powerful microscope. Instead of seeing a chaotic jumble of cancerous cells, you witness a battlefield where cancer cells are being systematically eradicated. The tumor mass shrinks, and healthy tissues begin to regenerate. This is the compelling evidence revealed by histopathological studies. They confirm the anti-cancer effects of Clostridium sporogenes, endospores, and Boswellic acid, painting a picture of hope for the future of cancer treatment. Section 10, a beacon of hope, the future of cancer treatment. The journey of scientific discovery is often long and winding, paved with both challenges and triumphs. The research on Clostridium sporogenes, endospores and Boswellic acid stands as a testament to the power of innovation and collaboration in the fight against cancer. These findings have the potential to revolutionize cancer therapy, offering a ray of hope to millions worldwide. Imagine a future where cancer is no longer a death sentence, but a manageable condition. A future where treatments are more targeted, less toxic, and more effective. As we delve deeper into the intricacies of cancer biology and explore the vast potential of natural compounds, we move closer to that brighter future. The universe of cancer research is vast. With each new discovery, we take another step towards a world free from cancer's grasp.
As we wrap up today's episode, I just want to take a moment to acknowledge the incredible work of some truly inspiring scientists. These individuals have dedicated their lives to pushing the boundaries of what we know about cancer and how we can fight it. Their tireless efforts and groundbreaking discoveries are paving the way for a brighter future. This research wouldn't have been possible without the dedication and brilliance of Professor Dr. Hany Ahmed Marghany Mahgoub, a professor of plant biotechnology at Al-Azhar University. His innovative approaches in plant biotechnology have provided new insights into cancer research, showing us that solutions can come from the most unexpected places. And Dr. Mostafa Ahmed Hussein Elbahnasawy, a lecturer of immunology at the same institution, and a postdoctoral fellow at the National Institute of Health in the United States. His work in immunology is crucial, as it helps us understand how the body's own defenses can be harnessed to fight cancer more effectively. Also, let's not forget Abdullah Samir Syed Ahmed Mohammed, who has been making strides since his B.Sc in microbiology in 2018 and his pre-master studies in 2020. His early contributions are already making a significant impact, and we can only imagine what he will achieve in the future. And last, but certainly not least, Dr. Rokhaya El Sayed Maruf Shams El-Din, a lecturer of biochemistry at the Egyptian Atomic Energy Authority. Her research in biochemistry is opening new doors to understanding the molecular mechanisms of cancer, which is essential for developing targeted therapies. These dedicated researchers are truly advancing our understanding of cancer and opening doors to innovative treatments. Their collaborative efforts and relentless pursuit of knowledge are what drive progress in this challenging field. Their work gives hope to millions and brings us one step closer to a future where cancer is no longer a devastating diagnosis. The impact of their research extends far beyond the lab, touching the lives of patients and their families around the world. But let's not forget, this journey isn't just about the scientists. It's about the collective effort of everyone involved, including the patients who participate in clinical trials and the communities that support this vital work. It's also about you, our amazing viewers who fuel this quest for knowledge and discovery. Your curiosity and support are what keep this channel and the scientific community moving forward. So, what do you think about this groundbreaking study? Your thoughts and feedback are invaluable to us, and we love hearing from you. Could their findings pave the way for future advancements in cancer treatment? The potential is immense, and we are excited to see where this research will lead. Let me know your thoughts in the comments below. Your engagement helps us create better content and fosters a community of like-minded individuals passionate about science. And hey, if you enjoyed this video, don't forget to like, subscribe, and stay tuned for more exciting research updates right here on For Science Salah Lotfy channel. Your support means the world to us and helps us continue bringing you the latest in scientific discoveries. Thanks for watching, and until next time, keep exploring. Stay curious, stay informed, and keep pushing the boundaries of what we know.